Okay, thank you. Thanks to the organizers for putting together this wonderful conference and giving me the chance to present our work. I also changed the title of my talk. So I'm going to talk about a protein that we found that regulates the assembly and function of cardiac dyads. So we've heard a lot in this conference about cardiomyocyte maturation. Of course, this is trying to understand how you get from a fetal or neonatal cell like this to an adult cardiomyocyte like this. This involves changes in many different aspects of the gene expression and function of these cells. So why is this important? Of course, many of us here are concerned about the immaturity of stem cell-derived cardiomyocytes since they get stuck in a fetal-like state. We've also heard about how dedifferentiation and redifferentiation are important for cardiac regeneration. We also think that abnormalities of maturation underlie some forms of cardiomyopathy or the adverse myocardial outcomes in congenital heart disease. So this one slide will summarize the work on transcriptional regulation that we did in cardiomyocyte maturation. So we tried to understand the hierarchy of maturation in terms of the different subprograms, such as metabolism, sarcomeres, ultrastructure, size, transcription. And we came up through doing many knockouts, many of them CRISPR-mediated knockouts. We developed a hierarchy of relationships. So on the top, of course, since I'm interested in transcription, transcription's on the top. We identified a epigenetic regulator, RNF2040, that seems to be important for the maturational shift in gene expression. We also found, of all the cardiac transcription factors we screened, that SRF had a uniquely important role in driving cardiomyocyte maturation, probably because it organizes sarcomeres. This is a bidirectional communication in which sarcomerogenesis regulates the abundance of filamentous versus G-actin, and that regulates MRTF. Of course, this is well established in smooth muscle cells, but interestingly, it had not been investigated in cardiomyocytes, and we showed that this pathway also functions in cardiomyocytes. We also found that SRF controls mitochondria and metabolism. But interestingly, none of the manipulations we've done to mitochondria have affected the ultrastructure of cardiomyocytes, which is a quite different result than what's coming out in the studies of iPS-derived myocytes. We found that sarcomeres are on top of the regulation of other phenotypes of the cell, such as the ultrastructure and actually the growth of myocytes. But in this talk, I want to focus more directly on one of the hallmarks of myocyte maturation, which is the cardiac dyad. So I originally got interested in dyads mostly because we were using them as a marker of maturation, but these are really amazing structures. So just to review, so everyone's on the same page. So we have the T-tubules that are invaginations of the plasma membrane. And then this membrane system is immediately opposed to the junctional SR, which is a specialization of the endoplasmic reticulum. When the action potential comes down the T-tubule, it activates the L-type calcium channel, which lets a little bit of calcium in, and then through calcium-induced calcium release, the SR releases a lot of calcium, which then goes to activate the sarcomere. So in mice, this structure, the dyad, develops at about P14, and it's, you could think of it as the heart of excitation-contraction coupling. So sometimes in those cartoons, you lose sight of what the actual structures look like. So this is a more realistic three-dimensional cartoon showing the plasma membrane with the invaginations and then the adjacent sarcoplasmic reticulum, which you can see on this EM. Here's the Z line with the T-tubule, and then immediately opposed to it is the junctional SR. And again, I want you to notice that the T-tubules are really tightly adherent to the junctional SR, and that all of these are aligned at the Z lines. So again, sometimes you lose sight of these things at those scales, and the light microscopy really shows the amazing organization of these structures within a cardiomyocyte. So here you can see the alignment of the Z discs to the junctional SR, and then the alignment of the T-tubules with the junctional SR. So sarcomere, T-tubule, junctional SR, all in perfect register. So the question is, how does this happen? This is really an amazing cell developmental feature of these myocytes, but actually very little is known about how these develop. So one protein that's been well known is BIN1, which is important for T-tubule curvature, formation of T-tubule microdomains, and L-type calcium trafficking. Another well-known protein is junctophyllin-2, which anchors to the junctional SR and has a binding domain that also attaches to T-tubules, so it helps to keep these two things opposed to each other. But this knowledge is mostly focused on how the T-tubules are organized. But in our literature search, we really couldn't figure out how the junctional SR is organized or what positions it next to the next to the Z line. And in fact, the junctional SR is positioned independently of the T-tubule. So we can look at myocytes that naturally do not really have T-tubules. So for example, atrial myocytes have few T-tubules, but they still have highly organized junctional SR. The same with a neonatal ventricular myocyte, also doesn't have T-tubules, but it still has organized ryanodine receptors. And if you go back to E15.5 cardiomyocytes that are still assembling their sarcomeres, you can see that when the ryanodine receptor is lined up, it's always next to a Z line. But you very rarely see a linearly organized ryanodine receptor without a Z line. So this suggests that the signal is coming from the Z disc to position the ryanodine receptor. So just to summarize, we have the Z disc somehow positions the junctional SR, but how that happens is not known. And then the junctional SR provides information for the assembly of the T-tubule, which is probably through junctophyllin and BIN1. So why is dyad assembly a significant question? In addition to just the biological interest in how this structure forms, it's well known that dyads are disrupted in many different types of heart disease, and it's thought to underlie the abnormalities of excitation-contraction coupling. So here's a mouse ventricular myocyte. There's a control, and then in a TAC model, you can see the disassembly and disorganization of the T-tubules. And this also applies to the junctional SR, which also becomes disorganized in heart failure. Another reason is that iPS-derived myocytes, we are well aware, don't make T-tubules very well. But I think it's maybe less well known what happens to the junctional SR in iPS-derived myocytes. So here we've micro-contact printed a iPS-derived myocyte so that it has a rectangular shape, and this really helps the cells assemble their sarcomeres so that they have well-organized Z discs. But even with this cue, you can see that the junctional SR is not organized. So there's something wrong with these cells that the Z disc cannot position the junctional SR, and so it's no surprise that they don't have T-tubules. So we undertook a proximity proteomic screen to identify proteins that are in the dyad. We took advantage of the known ryanodine receptor complex, which also involves these proteins junctin and triadin. And we fused the bio-ID to these proteins, and that will in vivo biotinylate proteins that are in the dyad. We then purified those proteins on streptavidin beads, and here's a summary of the results. We became very interested in this protein CMYA5, which was one of the top hits in this screen. So we validated that CMYA5 is in close proximity to the ryanodine receptor using a proximity ligation assay. So probably most of you have not heard of this protein. It's also known as myosprin. It's a very large protein, almost 4,000 amino acids. It has this highly conserved trim domain on the C-terminus. Most of it is poorly conserved and unstructured, and then it has a conserved N-terminus. This is selectively and robustly expressed in the heart and skeletal muscle, but there's been very few functional studies of this protein. One study that caught our attention was this yeast-2 hybrid screen done a few years ago, which showed that it interacts with the ryanodine receptor through this domain. They also showed that expression of this protein could change the clustering of the ryanodine receptor. So we made a knockout mouse of this gene, and we showed that it results in disassembly of the T-tubules and a disorganization of the junctional SR. Here's some electron micrographs showing the T-tubule. You can see it's shifted in its position from the Z-line, and the junctional SR has a very abnormal morphology. Wow, I'm really out of time here. We looked back at these myocytes that lack T-tubules, and we found that it has the same effect whether there's T-tubules or not. It disorganized the junctional SR. You could see that in embryonic wild-type myocytes that whenever the junctional SR was aligned to the Z-line, you could find CMYA5 at the Z-line. I'm going to skip this for time. So just to summarize, we think that this protein CMYA5 binds to the Z disc, and it tethers the junctional SR to the Z disc. Junctophyllin then reads the position of the junctional SR and assembles the T-tubule. Because of this protein-protein interaction between CMYA5 and the ryanodine receptor, we also wondered if it might directly regulate the activity of the ryanodine receptor. So we looked at calcium sparks, which are elevated in the mutants. We also looked at the calcium transients, and instead of this very nice sharp upstroke of the calcium transient, you get a ragged edge so that the calcium transient has a lower amplitude. We also found this interesting phenomenon in which, so when you have pacing spikes, each dyad should fire reliably at the pacing spikes, but we found many examples where dyads would sometimes fire and sometimes not in response to pacing. So we called that uncoupling and found that uncoupling was more frequent in the mutants. And at the organ level, the mutants had moderate and progressive LV dysfunction, and they really fell apart with pressure overload. Here you can see that under pressure overload, they had a severe ventricular dysfunction and highly abnormal calcium sparks. So to summarize, dyad assembly happens in a hierarchical manner in which the Z-line provides the cue to assemble the junctional SR, which then assembles the T-tubule. We think that CMYA5 is an important mediator of this positioning of the junctional SR to the Z-line, and that it directly regulates the activity of the ryanodine receptor. This protein is important to protect the heart from pressure overload. We find this very interesting because we think that there may be roles for gain of function of this protein to protect dyads from heart failure or to help to organize dyads in iPS cardiomyocytes. And we wonder if modifications of this protein may contribute to the disassembly of dyads in heart failure, as is seen with proteins like junctophyllin. So just thank everyone in my lab that did this. This was actually the work of a single postdoc, Fujian Liu, and we had many collaborators that helped us. Thank you.